picture of it for me.
Okay, so this is the motor neuron coming in, right? And this is the synaptic end bulb. As the action potential travels down here, there's a change in permeability, and that opens up sodium gauge, closes, and later, and sodium rushes in. Later, gates open for potassium, potassium rushes out. And that change in permeability is what we call an action potential nerve impulse. When it gets to the end, that change in voltage then across the membrane opens up the gated channels for calcium ions. Calcium rushes in because it's in high concentration outside.
And it goes to the
and it causes
right?
these vesicles shown in white here with their neurotransmitter molecules in them to fuse here and release the neurotransmitter by exocytosis. And those acetylcholine molecules then fuse across that gap here called the synaptic cleft. So this is neuron and this is muscle. This is called the motor end plate part of the muscle. And it doesn't show it here, but there's little receptors here for acetylcholine. When the acetylcholine binds to the receptor, that receptor also is a gated channel for sodium ions. It opens up, sodium rushes in, depolarizing the membrane. Later on, potassium channels will open up and potassium will rush out, repolarizing them. But that change in permeability travels along the membrane. When it gets to a T-tubule, it goes inside. So the T-tubules are really extensions of the sarcolemma and surround all the myofibers. These are myofibers. So it goes all the way inside. Okay. So the blue here are the T-tubules. When it gets inside, that change in permeability and everything, change in voltage, then that opens up the terminal cisterns, and calcium ions are released that were stored here in the terminal cisterns. This combination of two terminal cisterns, or the sarcoplasmic reticulum, plus the T-tubule is called a what?
Triad.
Triad. Okay. And you can see the bands here, etc. I think they mislabeled them in the video, though. I mean, there's actually another. Now, this would be what?
Sarcolemma.
Epimycete? Perimycete? No, it's epimycete. It's not the sarcolemma.
Perimycium?
Perimycium?
Endomycium. This is part of a single cell.
Damn
this
it.
is not a fascicle.
Endomycium.
Perimycium is around the whole fascicle. Epimycium is around the whole muscle. Okay. So this is just the sarcolemma, a single part of a single cell with the endomycium around it. Okay. So, let me hand this over here.
That's the $1,000 one?
Yeah. This is when I had the meeting. In case I got the little man. So this would be what?
T-tubules.
T-tubules.
Yeah.
And these would be the terminal cisterns, right? These would be a triad. That's already getting bumped up. So this is getting kind of screwy. I gotta get it restable. Okay, what's this called?
Z-disc.
Z-disc. Okay, it looks like a Z line, so like a Z-disc. What's the blue?
Myosin.
That's
Myosin.
myosin.
It makes
Thick.
up what? Okay, the thick filament, and this was the A-band.
A-band.
Okay. What's this? Thin filament. And what's in the thin filament? What's the
Actin.
red things?
Troponin.
What's the red?
Troponin.
Huh?
Troponin.
Globular actin.
G-actin.
G-actin.
Damn it.
When you put it all together and
But that was
twist
the
it,
one
it's
that
called
needed the calcium.
fibrous actin. You put fiber. What's the green?
Troponin.
Troponin, okay. Or some people say troponin. And what's the yellow?
Tropomyosin.
So the calcium that's released from the terminal cisterns here binds to the troponin.
Yeah.
Troponin has three parts to it, as you can see. One part binds calcium ions, the other part's attached to the globular actin, and the other part's attached to the tropomyosin. When the calcium binds, it changes shape, it pulls on the tropomyosin, uncovering the binding sites, or active sites on each little actin. Now the heads here are cross bridges of the myosin can glom on, swivels, ATP binds, it attaches, ATP is split, and it recocks itself. And it keeps doing that over and over again. So you die. Okay. So where's the A-band? Right, wherever there's myosin. So that's the dark band, DA, arc A. Where is the I band?
The space between the Z.
Okay, wherever you just have the thin filament. In the middle of the A band, we have the
M.
H zone, where there's only thin filament.
M band.
This is the
M.
M line.
M
It's
line.
actually several little proteins. It's not shown very well here. What's not shown here? The elastic filament.
Oh, the Titan.
Made of titan or connectin, it's called sometimes. And that's, supposing I read different things, it sort of stabilizes probably the A band, the myosin. Okay? So besides myosin in the thick filament, what else do you have? It's not shown here. And ATPase. This splits ATP.
And that hangs out where? In the myosin?
It's in the thick filament. Okay? So as the muscle contracts, okay, things slide together, right? Overlapping. So the thick and thin filaments do not shorten, they just slide past each other. So that's the sliding filament theory. So what's happening to the A band?
Stays the same.
Stays the same. What's happening to I band? It shortens and
Shortens.
disappears. What happens to H zone? Shortens and
Shortens,
disappears. These
disappears.
may actually overlap, okay? What happens to the sarcomere? Is that between here and here?
Z disc.
Okay. Okay. Is that okay?
Fantastic.
Okay. Make her away.